draw the topic for the day really quick, and then we'll do our like hellos and see how everybody's doing and kind of get into it. So, as always, I've got the old plague doctor mask full of ideas. Let's draw a ridiculous idea out of the hat. Let's pick a good one. I'm hoping this one's fun. I have no idea what this is. Okay, let's see what we got. This is gonna be fun. It says real toilet paper moonshine. So for those unfamiliar, um, my friend Nared made a video fairly recently where he made moonshine out of toilet paper. And the way he did it was he added cellulose to toilet paper and then like digested it and to get it down to actual like sugar, and then actually fermented it and then distilled it and made moonshine. So I would think that, I mean, this one should be real easy. It's just cellulose, but so I might actually end up doing a second one if this one's really fast, but we'll see. So yeah, it's basically I'm gonna probably do the same sort of thing that we've done in every other stream so far, which is make a plasmid for modifying yeast. So I think this should be fun. We'll get her done. So how's everybody doing? Hope everyone's staying safe and healthy and happy and all these things, or not going entirely stir crazy. Personally, part of the reason I'm doing the stream today, other than I just like streaming and I like hanging out with you guys. I think it's all good fun. Is I'm actually stuck waiting for some bio stuff to be done. So, that's exciting. I spent basically all of yesterday doing work on the Spider Silk project. I know everybody and their mother has been asking me about the Spider Silk project. And so yeah, it's not that the lack of videos is not because I haven't been working on it. It's because I've been working on it tirelessly and it's very hard. So the thing I was doing yesterday was, so basically I've been having a problem with the spider yeast, which is to say that actually getting the DNA into the yeast that the DNA was built to work in has been very hard. And I'm not sure why. I've been, following the protocols to the letter, and they just don't work for some reason. I think part of this is the species of yeast that the DNA is built to work in was not the species that I wanted. It was just the only thing that was available. So that species is called Pichia pastoris, and I much prefer working in Saccharomyces cerevisiae. So Saccharomyces is the sort of yeast that you get. It's like baker's yeast or brewer's yeast. Whereas Pichia is more of a spoilage yeast, so like it'll still grow in all the same things, but like you wouldn't want to eat it. It can like eat methanol. It's a really interesting organism, but it also grows much more slowly and is much more difficult to transform. Don't really know why. So I've been having issues getting the DNA into the yeast, so I figured, you know what, if this is becoming such a problem, I'd really like to be able to make the video and make an update and do all that kind of thing. But the only way that I'd be able to do that, if I'm stuck on this step, is if I build a new plasmid that's built to work in E. coli. So what I was doing yesterday was some of that work, basically. So we've got and I'll show all of this when I switch to screen share mode in a second. I'll show you what I've been doing, basically. But the idea is I took the DNA that the synthesis company made for me, that's built to work in yeast, and so basically all of yesterday was trying to clone out just the silk sequence so that I can then take it and stick it into the E. coli plasmid. So that was really, really frustrating. Because of how GC rich the silk sequence is, it's apparently just very difficult to PCR, and it's also very repetitive. So one of the issues that I had was I was getting a lot of like off-target garbage, basically. And if you go over to my Instagram page, which there should be links in the description. If you go over to Instagram, I've posted some pictures of those gels, like the results of this. So what I ended up having to do was first run a restriction digest. So literally like cut the basic chunk of the DNA out. And again, I'll show this on the thing in a second. But so I had to like cut out the chunk that I wanted with like, some extra-ish on the ends. And then I could actually go and run the PCR reaction, and it worked. And I don't know why that fixed it, but it did. So yeah, it was a lot of, it was very frustrating. It was a whole day worth of like run a gel, run a restriction, run a gel. That didn't work. Run another restriction, run another gel, run the PCR, run another gel. Like it was like a solid like 12 hours of doing stuff. So end of the day, I did end up managing to clone out my silk sequence, which is really good. Now I should be able to take that piece, take the E. coli plasmid that I want, and just like boop them together, and I should have a finished thing. So the thing I'm waiting for is actually for bacteria to grow. I need them to grow a certain amount so that I can actually take that finished DNA, put it into the bacteria, and then I should be able to, right then I should have silk immediately. Like if everything assembles properly, I will have silk by tomorrow. If it didn't, not so much. So, that's going to be interesting. But yeah, so that's what I've been working on. That's what I'm waiting for. So I figured a couple of hours of hanging out and designing some cool DNA and all that kind of stuff sounds pretty good. So, that'll be fun. I'm seeing a lot of questions in the chat about the human neuron project, as I've addressed in every stream so far. If you'd like to see the follow-up to that video, it's not on my channel. It's on Ben's channel on Applied Science. Go watch. I don't think it's his latest video anymore, but he made a video about making like photolithography masks using like an old like an antique like large format camera. That's the second part of this. Once the pandemic is over and I can actually, travel to the States and like do all these sorts of things, I'll be taking the thing that he makes and growing neurons on it. So he's now making the base plates and the preamplifier for actually picking up the signals from the neurons. And then yeah, I think that'll be a lot of fun. But yeah, pandemic has to kindly fuck off before I can actually do that. But yeah, so I think that'll be fun. Anyway, let's get into screen share mode so I can show you what we're working on today. Also, did it work? No, okay. There we go. Okay, so now we're in screen share mode. Wonderful. You may notice that once again there is a donation bar. This time, rather than raising funds for one of the plasmids, because I don't think I'd ever actually make this. Toilet paper moonshine sounds very fun, but there's already an organism, and I'll talk about this in a moment. But there's already an organism that you can just use directly for this. Like, there's no reason you genetically modify anything for this. But what the fundraiser is for is actually for a piece of equipment that I've been putting off buying for a very long time because it's expensive. But I've kind of hit the point where it would really make my life a lot easier to have it. It's called a qubit, or a qubit fluorometer. And basically what it does is you add a small amount of dye to a solution of DNA, and then you put it in the machine, and it'll tell you how much DNA is in the solution. And this sounds very trivial, but it's actually ridiculously important. So yeah, all the donations, and so I've got the fundraiser to run for a week. So for this week's streams, and there's gonna be two. There's gonna be one today, and I'm thinking one on Friday. I'm gonna be raising a little bit of money to try and make the price of that suck a little less. But yeah, so I'll be getting a qubit soon, and you'll see it show up in the videos, and it should make doing a lot of these steps just a lot easier. Like, knowing how much DNA you have just makes working so much easier. Like, you can actually know, like, okay, I need exactly this amount of DNA, this sort of thing. The actual cost of the qubit is about $1,200. I've got the fundraiser for $800 because I'm more than happy to just cover the whole cost myself if I have to. But I figure that's a reasonable chunk to make my life a lot easier and more comfortable. Yeah, unfortunately, bio equipment is fucking expensive, and there's just no good way around that. Unfortunately, I made a video a little while ago about like a DIY nano drop style spectrometer, and it's just not very good. Like the people who designed it, like I just used somebody else's design, so I had something as a baseline. But the people who designed it were not physicists. Like they were going for making a thing that looks very pretty, but they're not physicists. So it was designed very poorly internally, and so it doesn't actually work very well. So that was really frustrating. What am I drinking? Just some chamomile tea. But yeah, so that was really, really frustrating. And so yeah, at this point I've kind of hit a wall where I'm like, I could attempt to DIY another one, but I mean, access to materials has all of a sudden become very difficult. And also I may end up just spending a bunch of money on making a thing that ends up being crap. So it's easier to just be like, you know what? Let's just buy a thing that works. And I know that works. So yeah, that's the... Ooh, exciting. Thank you, Nico. You are forever da bomb. Much appreciated. Much, much appreciated. Thanks for getting that started. Okie dokie. So let's talk about toilet paper moonshine, as that is the topic for the day. It's funny, I just put that one in the hat yesterday, and I remixed the hat right before we started just to make sure that I didn't, like the older ideas weren't getting sunk to the bottom, because I mean it's a pointy ass hat, so things fall to the bottom. So yeah, that's yeah. I'm kind of surprised. It's funny, there's a few others that I put in there yesterday that I was kind of hoping that I'd draw just because I was excited about them and they sounded funny. But we'll see. Oh, yes, thank you for the reminder. Got to change the name of the stream. I'm gonna do that really quick and then we'll get into it. Okie dokie. That has been saved. Thank you for the reminder. I'm a turnip and I forget these things regularly. Glad we actually got to that within the first couple of minutes. Okay. So, with that done. Oh, thank you, DNA Farm Boy. Much appreciated. Why is that playing out of that speaker? There we go. Weird. Okie dokie. So, so toilet paper moonshine. So basically what's going to have to happen, the yeast will do the fermenting themselves, right? But so really what we need is something that's going to, whatchamacallit, break down the cellulose. So cellulose is really just a long chain of sugars, which is... Ooh, thank you, Shreyas. Thank you very much. Yes, Nile Red did already do this, but Nile added the cellulase separately. So I like I said, I'm gonna talk about how this works. So the thing that Nile did, which was very cool, by the way, was he added cellulase, which is the enzyme that breaks down cellulose. So if we look up what cellulose looks like, even though you can kind of see here, I want a nice big picture so everybody can see. And no, this won't be another Nile Red collab. Oh, thank you, Mr. Roberts. Wow, my internet has just ground to a halt. That's exciting. Bugger. Come on, work, please. Is it frozen on this side too? Oh, somebody did the thing. Thank you. Very much appreciated. I mean, stream's still going. It's just this page is not loading, and I do not know why. I'm gonna try opening it again. Is it just? Yes, I remembered to update Chrome. Thank you for the reminder. Why is this lagging so hard right now? This is annoying. Huh. This is awkward. So I'm gonna just since the internet's not cooperating, I'm gonna let that load and I'm gonna answer some questions because it's how we do. So, how's everyone else doing? How you dealing with the going stir crazy and the whole pandemic ish? Oh, did it load? Yay, it load. Okay, finally, we're back. Okay, so this is what cellulose looks like. That's a really tiny picture. This is a much bigger picture. Wonderful. Okay, so as you can see, this is a sugar molecule. It's literally glucose. It's like the most basic kind of sugar, and it's just a really long chain of them, like so. So you've got these, like, it's this really long chain of sugar molecules. And so what we need to do to be able to make this fermentable is we need to cut these into little pieces. The thing is, cellulose is a very tough material. Even though it's just made of sugar, like chemically, it's very, like it's just a very tough molecule. So you kind of have to, and also it packs really tightly. So it's not just like in like a cellulose fiber, right? Like in like, a bit of wood. It's not just one strand. It's thousands and thousands of thousands of these, and they crystallize. And it's sort of weird to think about wood as being a crystal, but it is. Like the strands sort of like interlock between them, which is sort of what this is showing, I believe. I'm going to see if I can cellulose hydrogen bonding. Oh, no, not peroxide. I want bonding. So yeah, basically what ends up happening, and you can see it in this image, is all the different chains of cellulose all like link together. So you kind of have to not only break the individual things apart, you got to break the strands apart too. So in nature, the way that this is done, is you basically have, I think it's 5 different enzymes that each kind of do this one step at a time. So like one of them pulls the strands apart, one of them breaks it into smaller pieces to like expose the ends, because the enzymes only work on the ends of the fibers. So if it doesn't break into smaller pieces, it's just like it enzymatically, it's just very slow. So I think that's part of what Nigel did, was he first broke the cellulose down a little bit before adding the cellulase. So where does cellulase come from? So let's look up cellulase just so you can see it. So this is cellulase, and of course there's many of them, and I believe this is the thing that I was talking about. Yeah. So okay, there's 3 in this picture, but so it goes endocellulase, which like breaks the chains apart. Then you've got exocellulase, which breaks the like the long chains into smaller pieces. Oh, thank you, Naldi. To answer your question, no, I am not still lactose tolerant. Anyway, yeah, so once the chains are broken into smaller pieces, then there's actually cellulobiase, which breaks it down into sugars. And once it's in sugars, the yeast can just ferment it normally. Like they don't care what kind of, like technically this is a different form of glucose. I think there's like alpha glucose and beta glucose, I think. And cellulose is made of the kind that we don't eat, but the yeast don't care. To them, it's all just yeast. Like it's all just sugar. They don't care at all. So where would you get these enzymes from normally? Now, one of my favorite organisms is called koji. And if you have not heard of koji, you must, because, oh boy, is it just a wonderful, wonderful organism. Also, if you have not picked up a copy of the Noma Guide to Fermentation and you're into ferments and microbes and that sort of thing, I'd highly recommend it. It's one of my favorite books, like cooking books. It's written by the chefs who run the restaurant Noma, and Noma is one of the best restaurants in the world. And one of the things that they talk a lot about is koji. And so koji is actually a type of fungus, like it's a mold. So you can see it here. It like if you didn't know that this was food, like you wouldn't think that this is food, but it is. And it's a type of mold, and normally it's grown on rice, but you can grow it on anything. Like it can break down like polysaccharides, so like things like cellulose or starches. It can break down lipids, it can break down proteins, and the ways that it does this are really, really fascinating, and also provide fantastic flavor. So if you've ever had soy sauce or miso or sake or a variety of other foods, they exist because of koji. So like rice, for example, is a very tough starch to break down. So you can't actually just ferment it. Like if you just grind up rice and throw a bunch of yeast at it, like nothing's going to happen. Like you'll get a little bit of fermentation, but like nothing really. So what you actually have to do is you have to add koji to your rice before you add the yeast. And there's all kinds of tradition on like how you do this exactly right, but frankly you could just like yeet it all into a barrel and it'll do the thing. It's a fantastic organism, and one of the things that it produces is cellulase. So a video that I'm actually working on right now is producing, and to the people asking about the lactose update. This is what the lactose update is about. So I'm going to be using koji, and I've actually got it fermenting just over there. I'm using koji to produce the lactase enzyme. And then I'm just going to show that because that's how it's done commercially. Like every time you've had a lactase pill, if you've ever had a lactase pill, they just grow literally like gallons and gallons and gallons of koji. Oh, somebody pitched in. Thank you very much. So yeah, they just grow gallons and gallons of koji and then like spin down the liquid and pull off the like spin it all down to get all the junk to the bottom. And then pull off the liquid and evaporate it, and that liquid is extremely dense in lactase. Like it just like exudes, like they grow it on lactose basically. So like media that contains a bunch of lactose, and then it just exudes it. But koji can also be used for making cellulase. So like a lot of the if you've ever seen them in like the health food stores, those like what are they called? Like the enzyme pills, like the digestive enzyme pills. Like most of those enzymes come from koji. It is an extremely versatile organism. It's fantastic for cooking, but also it's it just produces a huge amount of enzymes. So the cellulase that Nigel used, I believe, came from koji. So yeah, that's pretty great. So yeah, basically what we're going to need to do is we're going to need to emulate this process, but in yeast directly. And the nice thing is from the last things that we've done, like we're basically set and ready to do this pretty easily. So all we're going to have to do is find the appropriate enzymes. Oh, thank you for chipping in. Yeah, so the book is called the Noma. Here, I'll just look it up so y'all can see it once. And hopefully this isn't just lag city again. I really don't know why it's lagging so hard today, but it is. Chrome is garbage is I think the answer probably, or my internet's garbage. Not really sure which. But considering the stream is fine, I'm leaning towards Chrome is garbage. This is why I don't update things. Okay, yeah, this is gonna take its sweet fucking time. So while that loads, I'm gonna get the actual DNA stuff sorted. So we're going toilet paper moonshine. How do we know that the proteins will fold properly? We don't, but also it's pretty it's pretty rare that there's actually an issue with like proteins folding properly. Like it almost always works. Is this lagging too? Is everything just lagging? Is this just all lag all the time? I think it is. So yeah, this is the book. It's fantastic. Highly recommend. This is like if you know somebody who likes fermenting things and they don't have this book, it is a fantastic birthday present, Christmas present. Do you do presents at Easter? I have no idea. But yeah, it's a fantastic reference. Okay, good. We've got a new folder. Wonderful. And so I'm just going to import my empty Plasmid that I like using, which we found in the first stream. So, no, that's not right. Oh, man, it's in my other... Shit. Is it in here? It is not. Okay, well, so I'm going to have to actually import it from my other thing really quick, or maybe I'll just download... You know what? I'll just download it off AdGene again. 46815. This is... Yeah, okay, so this is my empty Plasmid that I like using. That was found by Sassy Tripper Sniper. Still don't know how to pronounce your name. Just call you Sassy. You're still fantastic. So this is it. This is basically like our empty program. Like this is like the library that we're using essentially. So it has most of the functions that we want, and so all we have to do is like you'll notice there's this cluster of stuff here. This is called a multiple cloning site. So it's basically an area in the DNA that's very easy to cut open and stick whatever you want in. So basically all we do is we would pay the gene synthesis company, and they would synthesize the gene, they would cut it open here, stick the thing in, patch it up, call it a day. So yeah, that's this. We're going to use this. I'm going to download it again. And I just realized that you download it by doing this. Let's download SnapGene file. Go back over to Benchling. And again, so this program that I'm using, we talked about last time. It's a web application. It's called Benchling. Normally it's my go-to for like taking notes while I'm working in the lab. It's really great for writing out protocols and just generally keeping notes, doing calendar stuff. But they recently added a whole bunch of like gene handling functionality, so I've been using it for that. And all things considered, it hasn't actually been that bad. It's mostly fine, which I appreciate. Okay, so it does not do drag and drop. That's kind of annoying. Choose file, downloads, add gene Plasmid. Exciting. Okie dokie. So now we've got our empty thing. Is it possible to do this as a 10th grade student with no money but having decent know-how? When you say do this, what do you mean? Do biology? Sure. I mean, anyone can ferment things. Anyone can brew and like work with organisms. But to do like genetic engineering, it is unfortunately pay to play. Like it's just every step is expensive. Well, thank you. I think that's meant to be King Julian, but it's spelled with a 1. Thank you very much. I very much appreciate the support. I'm just going to resize the chat a little bit so I can actually read y'all's messages. Okay, so I'm going to answer this question really quick. So he's asking, how does gene synthesis actually work? Do they use CRISPR? Okay, I'll start with the end. No, they don't use CRISPR. That's not how CRISPR works. And inevitably, we're going to end up touching on CRISPR at some point in this stream series because some of these ideas require it. But no, the way that they make DNA, like if they actually synthesize DNA, is what's called phosphoramidite chemistry. Ramidite. See if that's spelled right. And again, we're gonna have to just embrace the lag, which is exciting. So while that's loading, I'm gonna get this. I mean, this is all loaded in now, so I should be able to just close this. Let's copy this to just Spiderbrick main. So this way now I have a copy of this, so because I like doing non-destructive edits if I can. Oh, good, it loaded. Okay, so this is phosphoramidite chemistry. It's really nasty. Like, it's really nasty, but it is effective. So basically, what happens is you start with this. This is a phosphoramidite. So this is basically the DNA like backbone essentially, and then you've got your whatever like base it is. So like whatever letter it is, and all these little dangly bits basically let you selectively stick like a thing to one end. So like these are like protection and deprotect and groups that basically make it easy to do a reaction on one side while leaving the other end alone. And so you basically go in a cycle. This is the cycle. And so you start with your sugar with your base hanging off of it. You deprotect one end, add the next piece, cap one end, do a thing. Like it's just this big cycle of literally adding one base at a time, and you've got to like deprotect and reprotect and do all the reactions. And so doing this by hand would be insane. So it's done by a machine. So like you basically you'll see that it's got this little sphere looking thing here. That's not like a graphic. Like it's representative of you do this with these materials literally stuck to a bead, and either they move the bead between different solutions, or they just hold the bead down. Usually it's magnetic, and they just like flow the solutions over the bead one at a time. And so it's all automated, but like it's still a really frustrating process, even with the even with a machine doing it. Like just the humidity in the air can mess with the reactions. They're very sensitive, so it's not a great technique. So basically, and also it's limited. Like you can only do 200 bases approximately at a time before it starts making huge errors. So what you end up having to do is make like pieces that are about 180 base pairs, like letters long, and then putting all of those together individually. Thank you, Herr Dernacht. Yeah, man, usernames are hard to pronounce. But thank you very much. Very much appreciate it. Okie dokie. So yeah, that's how gene synthesis works, and it's a machine. I mean, I can show it to you. It doesn't really look like anything. It's basically just, yeah, it looks like a box with a bunch of bottles. And so each one of these bottles has one of the different chemicals in it. 4 of them are going to be the different letters, basically. And then the rest are going to be the actual reaction solutions that do the thing. And so what you would do, basically, is you give the machine a sequence, and you're like, make me this. And it just, like, does all the chemistry and then poops out a sequence that's exactly, well, in theory, the thing that you ordered. And this is why you pay other people to do this, because this machine's expensive, the chemicals are expensive, and it fails most of the time. So part of what you're paying for when you pay for gene synthesis is the guarantee that the thing that they made is what you ordered. So once they've made all the little fragments and put them all together, then they sequence it and make sure that they've got the right thing. Like if there's an error, they've got to go back and redo it, basically. And so that's what you're paying for. And, it's worth every penny. Cool. So this is now done. So I can close this, make this a little bigger, so y'all can see it a little clearer. So just as a review for people who are new to the stream, this big circle is called a plasmid. It's basically like an empty, or it's not an empty piece of DNA. These little tags basically tell you what the various letters code for. So if we click on this, for example, you'll see it just looks like a bunch of letters. Like this doesn't parse to humans. Like it looks totally random. So the whole point of having the labels there is so you know what each chunk does, basically. In this case, this red bit, or more specifically this blue bit, um, is a protein, and so proteins always start with ATG. That's literally the start codon. It's like protein starts here, um, and in this case it says CAN-R. CAN-R means it's kanamycin resistance. So it's a protein that will give the organism resistance to kanamycin. Oh, thank you, George. Very much appreciated. What's a good place to get started doing genetic engineering as a high schooler? If you can find a local iGEM team. iGEM is dope. Highly suggest. Otherwise, maybe ask your teachers, see if they're willing to set something up, or maybe make a little lab in your house. That's how I did it. Yeah. So start codon, kanamycin resistance. Yeast are actually naturally resistant to kanamycin, so you'd think that this isn't helpful, but the same gene that codes for kanamycin resistance also gives resistance to a variety of other antibiotics because they're chemically very, very similar. So, for example, this is kanamycin. You'll notice it looks like a bunch of weird sugars stuck together because that's basically what it is. But G418 is a derivative of kanamycin. You'll see that it looks very similar, but it's basically got some extra little functional groups on it that make it basically it's poison. Like, it's not even an antibiotic at this point. Like, you wouldn't ever use G418 on a person. We call it an antibiotic because it kills things, but like, it's poison. Like, it's not, you'd never use it in a medical situation. But yeah, so G418 is much more effective. And so you can actually use it on yeast, and it's coded, and like the resistance is coded for by the same gene. I believe the gene like breaks one of these groups. I don't remember which. But yeah, so the same gene codes for the same thing. So as long as we've got this CAN here, whatever yeast we put this into would be resistant to G418, and so we can use that to select them. So like once you put the DNA into the yeast, like how do you tell which yeast has taken the DNA and which ones haven't? Well, you put them on a plate that contains G418, and so only the ones that took in the DNA will actually survive. So there's some other ish in the plasmid, but we're not going to talk about it right now because it doesn't really matter. Now we're going to jump to this because this should get us to our multiple cloning site, and it does. So we've been cutting with Sal, but I mean small one also works. This TF1 promoter, this is basically the bit that this is sort of like the start of the program. So this tells the biochemical machines that like there is a gene coming soon. Like, so they bind here, and then they like scan along until they hit whatever gene we put in here. So now all we got to do is find some cellulase. Uniprot. So as usual, we're using a site called Uniprot, because it gives you the like amino sequence of like, it's basically just a database full of all sorts of different proteins and enzymes and stuff. So in this case, this is cellulase taken from Bacillus subtilis. So that's a bacteria. Shouldn't really matter. It should all do the same job. And luckily, it's actually nice and small. So this is, as proteins go, like this is a pretty small one. Are we seeing some fucking? By the way, if I see any like conspiracy garbage in the chat, like you're instantly getting banned. Let me just be really clear about that. Anyway, but yeah, so this is a really small protein as these things go, especially for an enzyme. Like, it's a really tiny enzyme. I'm not sure which cellulase this is though. I assume that this is the last cellulase. Like it's the in that chain that we looked at before. I assume it's the one that actually does the break the cellulose into sugars. So I'm going to just look up that pathway again, just so that I can actually see like what are the names, like just, refer to it properly, because a lot of bio is just like knowing the right names for things. I got a quick question. When are you going to revisit the GOES 16 Meteor satellite things? Eventually. I don't know. I don't know. I don't have a better answer than that, honestly. This is the one I want. Yeah, I'll do radio stuff when I can do radio stuff. At the moment, there's a pandemic, and the police do not take kindly to you hanging out in parks. So, is a $6,000 fine worth setting up an antenna and making a video? No, unfortunately. Um, Come on, this is so laggy. There we go. Now it's cooperating. What a concept. Yeah, okay. So interesting. Cellulose. So then what would the cellulase do? Yeah, I think cellulase is really the last step. It's the one that does the sugar. So we're gonna grab this. We're gonna throw it in, and we're gonna do the same P2A thing that we did in the first stream, just because it's effective. And I'm lazy. Copy. So now, moment of truth. Let's find out if I'm gonna use small one this time. Just for novelty. Pasty paste. Oh. Huh. Interesting. So this one, so evidently benchling does not have the same functionality as analyzes translation. No. So yeah, Genome Compiler, the tool that I liked using, is it like had a functionality that if you paste in amino acids, it would just automatically translate it to DNA. This evidently does not. So that's really annoying. Not sure what to do about that. Oh. There's also no control. Oh, there is a control Z. Oh God, what happened? Where'd it go? Okay, well, that was weird. Okay, so we're back at small one. So I might need to do the translation separately. Insert part. No, that's not right. Interesting. I mean, this is sort of the problem with using a new tool. What are the legal limitations in the U.S. on DIY hobbyist gene editing, on humans? If it's on humans, you can get into huge trouble. Otherwise, generally the rules are don't make anything infectious, don't make any toxins, and you can kind of do whatever you want. And also don't release it. And like you've got to be really careful, but like don't release it. Otherwise you'll get in trouble with the EPA. Don't sell it unless you've got clearance to sell it. Yeah, so it's... but for the most part, you can kind of do whatever. Why did this work last? I feel like this... Yeah, translation did work last time on benchling, right? Like I'm not going crazy. Like it did the thing. Why is it not pasting properly this time? I don't understand. This is very frustrating. Let's copy this again. See if maybe it's just... Is this like just slow? Pasty paste. Not all valid. Okay, it says it's working. Huh. That's really frustrating. There must be a character in here that it doesn't like. Or something. I really have no idea. I don't know why that's doing that. That's worse. Okay. New plan. I'm going to delete this. And no, actually, I do need to post it in. Why the hell? Could be that F it doesn't like. This is very strange. Yeah, it was the F on the end it didn't like. Okay, so let's just undo that. Hop over to here, grab everything other than the F. I don't know why they're... Never seen an F used. So maybe that's the thing that's freaking it out, I don't know. Okay, that should have, yeah. Alright, pasty paste. No, it still had an issue. Alright, I have no idea why it's having a fit. It's very strange. I've never had this issue before. But then again, I've also very rarely used Benchling. That's strange. That's weird. What are my options? Primers, history, alignments, CRISPR. There's a whole CRISPR section. That's neat. Analyze as translation. Okay, that's fine. Alright, well. Let's just do this. We'll just look it up. DNA, or no. Hopefully this is the right thing. No, this is not the right thing. I think this is the one that I used last time. Yeah, here we go. Okay, so amino acid sequence, pasty paste. Saccharomyces cerevisiae. Reverse translate, por favor. Oh, wait. T's. Copy. Okay, well, this should have fixed this. Very annoying. Don't know why it did that. But that should fix that. Pasty paste. Yay! Okay, good. Let's annotate this. Cellulase. Very exciting. Okay, we've got our cellulase in. Very good. Don't forget the F. Did I forget the F? Whatever, it's just one. What is, I can never remember my one-letter codes. I mean, fine. I'll throw the F in there just because people seem to care. Oh, it's literally just 3 T's. Okay, I can fix that really quick. No, I know. There we go. A collection of T's. Cellulase. Let's add 3 on here. That should fix that. I don't know if you caught that, but I did codon optimize. It was part of this tool, so I think I just did it too fast, and you might not have noticed it. But yeah, so Saccharomyces cerevisiae, it is codon optimized. And Benchling is now thinking real hard about its life choices. Oh, okay. Well, that was weird. Wait, what? Where did it... Oh, come on. That's exciting. It decided to crap out. So I might need to do that whole section again. Man, this is just not cooperating today, and I do not know why. I mean, I see the TTT there, so it should be fine. All right, I'm just not gonna mess with it. What's this go to? 246. Yeah, this really should not be such a hard thing to do. I should just be able to do that and click save and what? Okay. Wow, this is really just not cooperating today. Sorry about this. Alright, well, this is annoying, but I'm going to just stop messing with it before it shits the bed again, because this is just getting incredibly annoying. Okay, so we've got cellulase, which is the last step in this process. Let's find some exocellulase. Just look this up really quick. Exocellulase. This looks right. I just don't know. Oh, interesting. Okay, so these look more like the enzymes that I was expecting. So it says cellulobisidase, which I assume is that like last step in the. Wow, this is much larger. Let's see what we got for exocellulase though. Also, we're gonna have to do a couple little changes on the thing that we haven't done yet. So I just want to see how large this. Oh my God, that is way larger. It makes me wonder if that Uniprot thing, like the last thing that we used, was. Did I close it? I must have. If that was a fragment, if maybe I just grabbed something tiny, or if it is the whole thing. Yeah, see? I'm glad I caught that. So this is fragment. That's why it was so small. That is very frustrating. Okay, so now I actually have to basically start this again. So I'm going to just Control Z, just delete the whole thing. Come on, cooperate. Okay, we should be back to just empty thing again. Nope, it's still there. It's just not labeled. Now we're back to empty plasmid. Okay, well, isn't this frustrating? This is the fun of biology. Sometimes you just kind of get fucked. So this says complete. So this is the actual full protein. Which one is this one? This is the exocellulase. Oh, these are going to be huge. Okay, yeah, so this is definitely outside of, like, synthesizable. And again, because Koji exists, there's literally no reason to ever make this. But we're doing it because it's fun, and it's a learning experience. But as you can see, this is much, much larger. So that's one of them. So this is going to be our exocellulase. Then we need actual cellulase. So I'm going to go back. We're going to see what we got. As the lactose thing, we never thought it was going to be permanent. Like, the paper said it would last a minimum of 6 months, and it lasted at least that long. It lasted a year and a half. Seeing if there's any. Wow, this is just very laggy today. I really don't know why. Yay. Okay, good. This is actually loaded. So I'm going to go from maybe a different organism. because evidently the Bacillus subtilis thing just was not complete. Cell A. Let's see what this one is. Could be exciting. Some random organism. Looks more complete. So this one's called Cell A. So I just want to see what that actually is. Would it be possible to make a series of different yeast to make a whole diet? I mean, I don't know why you would want that, but I can't see why not. It would just suck. Like, this is one of those things. I never understood the Soylent crowd. Like, why do you hate food? Like, food is great. Like, I like my food to have some texture. Okay. Hydrolyzed microcrystalline cellulose. So I'm just reading through this, see what this is telling me. Close this so there's a little more room. Yeah, I just want to see what this is, because I mean, if this is just like one enzyme that does the whole job, then that would be like very effective and very helpful. Would you try CRISPR experiments anytime? Yeah, sure. I mean, CRISPR is neat. Like, I don't hate it. It's just, it's for whatever reason, that's the thing all the media talks about. But like, the number of times I've needed CRISPR, almost none. Like, I don't do stuff that ever really needs it. It's just not. It's like, I suppose when all you've got is a hammer, everything looks like a nail. But like, it's just not that helpful. I mean, it is for certain things, but like, people want to just throw it at everything, and I just don't understand why. Yeah, this is interesting. Like, this enzyme seems to do a lot of the different things that we want. I just don't understand. What is this? Where is this from? I'll go back, see what my other options were. See, there's like cellulase A, there's cellulase B. I don't know what these are and what the difference is. Let's see what cell B is. And this is kind of the research process. This is just, you know, just kind of just look things up, see how they work, check again, check again, check again. Include beta glucosidase. So cell B, like cellulase B, seems to be a very suitable model for blah blah blah. What else do we got here? Stable production. Okay, that doesn't help me. See, what's really interesting about this one, and I'm realizing that this is what I was looking at here, or maybe I'm not, but it looks like this enzyme is from an organism that lives in a very, very hot environment, which would make it extremely thermostable, which is neat, but I don't know if that's going to really help us. Let's see. Let's just look this up. Let's see how somebody's done this before, because frankly, this is just like doing this like random walk trying to find the right things is just not happening. So let's see if, here we go. See, this is always the best course of action. Like, start with a paper, like find a paper that does the thing that you want, and then work from there. Okay. This is neat. Okay, so this is basically exactly what I was going to do, just I was going to do it in yeast. So they've taken a cellulase enzyme and they've made it secrete from the E. coli. So, okay, here we go. So they're using beta-glucosidase was fused to cell C and D. So to me, what this says is they've basically made one gigantic fusion protein thing. Oh, somebody kicked in. Oh, thank you. Undingen? Probably mispronouncing that, but thank you very much. Greatly appreciate it. So yeah, from the looks of this, they've taken the one enzyme that knocks the sugars off of the chain and fused it to the enzymes that break the chains apart. So that's actually really handy. That's really, really handy. And also now I know the correct name to look up. So if I look up beta-glucosidase, I'd be willing to bet that the thing that comes up is the actual enzyme that I want, rather than... Okay, that's fine. I don't want to. What the hell? There we go. There we go. What the hell? Systolic beta-glucosidase. I did not know that humans make this. But we're going to go with one from Aspergillus, because Aspergillus is a fungus, and we want fungus. Hydrolysis of terminal non-reducing beta-glucosol residues with release of beta-D-glucose. So yeah, this is the thing that we want. This knocks the sugars off the end of the chain, which would make them fermentable. So we're gonna grab. Wow, that's a chunky boy. Okay, well let's grab the fastest sequence. So we have Z's. A little copy. Go over back to our reverse translate thingy. And I'm setting the output here to just one long line because I don't want it to like spit out a column. I want just one thing I can copy really easy. So we're going to go reverse translate, and then that should give us a nice long sequence of just obnoxious length. Let's do a little check, make sure that this looks right. That looks fine. Cool. So now we can actually copy this in. I just gotta find my spot. Okay, we're gonna do small one again. Pasty paste. Wonderful. It actually did the thing for once. So let's go create annotation. We're gonna call this beta glucosidase. I think. Is that what we called it? Was that the name of the thing? Glucosidase. Yes, that's correct. Wonderful. Cool. So we've got beta glucosidase. Annotation type. I don't know what that means. Save. And just so that we can see it, let's go forward. See if this does the thing I want. No, it did not. That is not even close to the thing that I wanted. There we go. Okay, this is what I wanted. Cool. So now it'll actually show the amino acids that this is going to make. Now, there's one thing that we're going to need to add before we move on to the before we move on to the next enzyme, which by the looks of it is going to be 2 more, or maybe 1 more if we can just find the fusion CD thing. Is we need to add a secretion sequence, and we're going to use the same one that we've used in essentially. Wow, thank you, Andrew Hill. Very much appreciated. Okie dokie. Yeah. So we're gonna throw in. We're gonna throw in a secretion tag, and so for that, I'm just gonna open up my projects, and we're gonna go to probably the milk and egg yeast thing. Now it's also saying that Benchling has a new version, so I'm gonna go ahead and refresh the page after I copy this, I think, and then we'll get to it. Will I be doing these streams every day? No, not every day, but at least twice a week. I'm thinking. Probably Mondays and Fridays is sort of what I'm thinking so far. But I'm open to feedback on that. I wonder if this is just okay. Well, this is not cooperating. So I'm gonna go ahead and refresh the page. I don't think I'm gonna actually wait. Cancel. I'm gonna okay. Save that. Hopefully that doesn't make it shit the bed, which it did not. Wonderful. Oh, this opened as well. Cool. So this is the plasmid that we made in the very first episode, and so I should just be able to grab this and I'll set it to plasmid view so you can see the whole thing. But what we made was basically this would make egg white. So that's what this plasmid's for. And we actually just ran a fundraiser in the last stream to to basically pay for the synthesis. So I'm going to actually have this plasmid made because it's small and it's sleek, and I think it will actually work. Cool. So this is the thing that we added at the very front of the plasmid last time. It's the alpha mating factor secretion signal. So I'm just going to copy it and I'll talk about it when I get back into the other plasmid that we're working on. Cool. So now I'm just going to paste it right at the beginning here because it's actually part of a protein. That looks very wrong. Something has happened and I don't know what. The hell? Oh, come on. Did it? Oh, I started one letter over. There we go. That should have fixed that. Why is this so large? That doesn't look right. That does not look right at all. Maybe it is. Yeah, no, it's a tiny little protein. Why is that? Yes, copy. Okay, well that's weird. That is very, very weird. I don't know why that happened. Okay, hopefully this is gonna go back. Close this. Control Z. Good. Did that fix it? Yeah, it did. Weird. Very weird. Okay, that should be the... Hey! It actually understood what I wanted this time. Huzzah. Yeah, so cool. Now we've got our secretion signal, and I'm going to actually set this to translate. Yay! Good, it did the thing. So you'll notice that this is actually a protein. Like this is a chunk of a protein, which the yeast would register as I need to secrete this. It's a mating factor. So think of this as like yeast sex hormones, but it's only a piece of it. So we take just like the front little chunk of it, and so the yeast recognize this as okay, this needs to leave the cell. But anything that's stuck to it, which in this case is going to be the beta-glucosidase, will also get secreted because they're stuck together. Like they're physically stuck together. One of the other interesting things that I noticed in this is because they're using a fusion protein means I don't actually need to bother with doing multiple proteins that have multiple secretion signals and all that kind of stuff. What it should be able to do is I should just be able to make it as one giant chunky protein that it just excretes, and I'll put a linker between them, and that should be fine. Will I ever do coding projects? Well, I suck at code, so no. Does the secretion tag modify the protein? Not really. It's essentially ignored. It's one of the... Oh, thank you, Hayden. Very, very much appreciated. It's basically just like a little gimpy dangly bit. Like it doesn't really affect the protein at all. This is sort of the reason that people use it. People are working on making smaller secretion signals, like see if they can do this in less letters, because I mean this is a bit chunky. But you know, you can do that. The other option is you can add what's called a cleavable linker. So it's basically like a little short string of amino acids that you could put like right here, like right between these two things. Um, why is that bell? Basically what would happen is once this is synthesized, one of the enzymes that's just like floating around on the inside of the yeast would recognize it and just cut it there. So this would get cut off, and you would end up with just the pure protein. But for something like this, it shouldn't matter. As somebody started a dangly bit drinking game, in which case you're going to be hammered by the end of this. So I think you've asked about the filamentous fungi in every stream thus far, so I'll go ahead and answer that. Maybe? I don't know. I have no intention of doing that at the moment, but if it comes up in one of the projects, sure, why not? Modifying filamentous fungi are a pain, though. It's not like yeast where it's just like one cell with a big nucleus in it. Like a filamentous fungi is hundreds of nuclei. It's basically like one big cell. Like it's weird, and modifying it's hard, and you've got to like use equipment which I don't have, and methods that are frustrating. And it's just like working with yeast is hard enough. Like trying to like throw some diesel on that fire of like, now let's also work with a thing with 1,000 nuclei per cell. It's not fun. Because if even one of the nuclei aren't modified, it can completely mess with your whole thing. It's not easy. You gotta like deal with spores and like modifying spores. It's frustrating. And no, CRISPR is not magic. It does not just magically fix bio problems. That's not how that works. Okay. Anyways, moving on. So we've got our secretion tag. We've got our first thing. So we're actually doing pretty good as these things go. So now we got to find the next two proteins, which I'm not going to use this one. I'm gonna start a new tab. Cell CD. Let's see if UniProt has it. They might. It'd be good. So you find all this stuff easier than coding. I mean, I code meat. That's kind of my... it's what I've been trained to do. It's what I've had practice doing. So it's what I'm used to. It's entirely different. Like, it's very little math. Okay. I like this note. Probably. It probably catalyzes. This is interesting. I don't know if this is the right thing. I'm gonna pull this paper up. See if I wonder if they've got a sequence in here. That would be really great. Is there supplemental information? No, there's just nothing. Let's see if it's at the bottom. Sometimes. Oh! What's this? No, just primers. Unfortunately, if you start seeing primers in papers, like the chance of this basically being made by like cloning out genes from an organism is pretty high, and I hate doing that. I spent all of yesterday fighting with PCRs. I hate PCR. It's awful. Did you get the Pro PCR from the Odin? It'll be a very cold day in hell before I ever buy something from the Odin again. Anyway, moving on. Do we have supplemental information with sequences? We do not. That's really frustrating. Okay, let's see the first time it mentions the cell D thing. Production of other hydrolytic enzymes. That's not helpful. In a previous study, we found that a catalytic domain of cellulase, cell CD, and its N-terminus can be employed as carriers for extracellular production of recombinant proteins. That is not helpful. We identify the catalytic domain of an alkaline cellulase with endo-beta-glucosidase activity, which sounds like the thing that we want. Okay, So it's in this other paper. So, okay, we go to the other paper. PubMed. What did the Odin do? They sell garbage. Is what the, yeah, like they sell garbage and like they sell lies, and I refuse to give them any money. Like I've had tubes from them show up empty. I've had like they literally sent me a vial of fucking water instead of DNA. Like I'd gotten their GFP yeast kit. Turns out the reason, like after doing it like 4 times trying to figure out like why isn't it working, yeah, it's because the tube that was supposed to contain 100 micrograms per microliter or 100 nanograms per microliter of DNA had nothing. Like it was straight water. Like some of the tubes of bacteria they sent me were contaminated. They sent me a tube of AMP at one point, which was just empty. And yeah, so they suck. Don't buy from them. Okay, this seems to be the new thing. Generally intracellular, that's fine. Results. We identified the catalytic domain of a cellulase from Bacillus subtilis, which is that thing that we looked at first. Cool. Very cool. So let's see where the full text is. Is it free? Huh, it is. What do you know? Will the fluorometer check for that? So will it check for DNA? Yeah, absolutely. It's even more sensitive than so we used a nanodrop, which is ironically a more expensive but less sensitive test for the presence of DNA. So we did 2 things. We ran that vial of stuff on a gel, which should show you even a couple of nanograms worth of DNA. Like if there was anything in there, it should have glowed really brightly. Didn't see anything. Then we ran it on a through a nanodrop, which can directly measure how much DNA is there. Again, nothing. Like below the threshold of the thing. But then we ran PCR on the sample, and we did manage to get a band. So like there must have been like a molecule or 2 of DNA in there. So I guess they didn't just straight up send water. But something about their purification or isolation of the DNA was just ass, and they just didn't check. So yeah, don't buy from the Odin. There's so many better places that, like, you know, some of them require that you set up a company, or even have a commercial address to buy from, but it's worth it. Like setting up an LLC in the States costs nothing, and is very easy to do, and it's worth it because then when you go to buy from a company, they treat you as professionals and they'll sell you whatever you want. There's lots of other entry-level kits. Carolina Biological sells a lot of them. Amino.bio sells a bunch of them. I mean, in my latest genetic engineering video, which I believe is called "These Bacteria Are Part Jellyfish," if somebody could throw a link to that up in the chat, that'd be great. But at the end of that video, I spent like 6 minutes or 7 minutes just talking about all the different places you can buy these things from. Use that as a resource. Don't buy from the Odin. Okay, so an LLC is $100. Whatever. $100 to be able to buy from any of these big companies is worth every penny. Also, biology is expensive. Like if $100 is the thing that's like stopping you from doing bio, find a new hobby. Like it's really expensive. This is it. Like it's just the way it is. Like and I talked about this on the last stream. Every single every single action you do in biology costs money. It's just the way that it is, and it's unfortunate, but it is. Okay, so this is the thing that I want, but does it contain the sequence for the enzyme that I want? And I'm going to go ahead and assume no. But it did say the name of the enzyme, so that was exciting. So I might be able to just look that up. It's secreted into the medium, cellular loculations. Thank you, James. Y'all, if you're shitposting in the chat, you're gonna get banned. Don't. Um, so as to the commercial address, that's actually easier to deal with than you'd think. I mean, like if you've got a friend who runs a business, you can just use that address. If you find your local like hackerspace and like are nice, most of the time they're totally fine with, and because they always have commercial addresses. So like, go to the hackerspace, make some friends, be like, hey, do you mind if I ship some cool bio supplies here? And they'll be like, yeah, sure, that's fine. Um, but I mean, it's worth it. Like if you sort those problems, then you're not worrying about buying kits from people who will just like sell you. Like first of all, Carolina will sell to anyone. Like it's Carolina Biological, so you can get great kits from them. They're a little pricey, but like they're very high quality and does the thing. Um, yeah, like UPS has business addresses. There's all kinds of ways to deal with this problem. Like if your answer to the problem is buy from a company that might sell you garbage, your money is already on fire. So you might as well invest in getting set up properly, so that you can do the thing properly and then not have a problem. Um, do you have any degrees or academical training? I do not. I do not have any degrees. I did a couple of years of bio in university, but I dropped out, and all of the things that we're doing today was not taught to me in university. Um, okay, what else we got? Yeah, so this is not. It just said the name of the thing. Where did it go? Hmm. It had said the name a little lower down of a. It just says a cellulase. It doesn't say which. Um, it could be this. I don't know, but it seems real small to be the thing that I'm looking for. We might just have to go with something else. Oh wait, did I just see a uniprot? Yeah, it was like endo beta something. Um, let's look up endo. No, that's endogenous. That's not helpful. Yeah, here we go. Wait. If I look up uniprot in this, no, that was. I almost thought we were gonna get lucky for a second there. Um, let's see if we can find this here. Oh, I'm seeing Vivek is given a uniprot thing. I will look that up and see what we got. Uniprot P10477. See, this is my favorite part about doing these streams. Like, I can sit and bumble around. I mean, and admittedly, I'd probably work faster if I wasn't like trying to be entertaining and also like chat and answer questions and stuff. Oh, this is perfect. Dude, Vivek, thank you very much. Very helpful. Looks like exactly the thing I need. Now, does it provide... Oh, yep, that's a chonky boy. But that is okay, because if it's only one chonky boy, then it's not so bad. Copy. Alright, let's go to reverse translate. Pasty paste. Reverse translate, por favor. Very nice. Wonderful. Okay, now we can go back over to here. Just grab this, because that'll get us to the end. Now, what I need to do is I need to put in a little linker, because I want to do a fusion protein. So this is something we haven't done on the stream yet. We've done P2A, which is a cleavable linker. So you get big backbone. Very funny. So in the first stream, and I don't have that open, but I can open it really quick. So in the first stream, we made this, which is the deer milk plasmid. So this would make a sort of pseudo milk thing from deer, and it's 4 proteins. Here, I'll close this so this is easier to see. Let's scroll to it. So you can kind of see it here. So it's 4 proteins. So casein, lactoalbumin, and lactoglobulin. And the way that we got 4 proteins off of one promoter, rather than having to add an individual promoter to every single one of these, is we used this, which is called P2A. P2A is what's called a self-cleavable linker, but that's not really what's happening. Like nothing's getting cut. What ends up happening is as the ribosome, which is the protein complex that turns RNA into protein, as it's reading this, it's like reading through and reading through and reading through and reading through, it hits this part and it kind of stutters. So it lets go of the thing that it was just making and like stutters and jumps, and then basically once it hits here, it starts making protein again. So by putting this in there, you can get multiple separate proteins off of one promoter. But I want to see. I think this time we're going to do something different. Oh, okay. Thank you, Gemma. Those are actually really, really helpful tips. Again, this is only my like second time using Benchling for this, so I very much appreciate the tips because I'm not familiar with it yet. Nate says you should alternate using use of the 2A to have more efficient use. Okay, I mean that's fair. I haven't submit. So we've done the funding for this plasmid, but I'm not 100% comfortable with it yet. There's some like little changes I wanted to go in. I'd been thinking about what happens if you use P2A over and over again. Yes, we're still doing the not milk. It's gonna happen. But yeah, so I wanted to go in. So there's P2A. There's multiple 2A linkers, right? So P2A was just the one that I used. There's T2A. There's a few others. So I was thinking of going in and just subbing some of these out with some of the other ones just to make sure that you don't end up with like any weird issues. And then there was a few other little changes that I wanted to make. But this will get made at some point. Anyway, not to get overly distracted, to do our thingy here. Where did it go? Okay, here. Yeah, so to do this, rather than using P2A, I want to just do a glycine linker. So I'm gonna paste in our gene, and hopefully that'll just work nicely. Paste, please. Pretty, please. Yay. Okay. Wow, that's even bigger than the last one. Is what she said. So this one was endo beta glucosidase, I think. I think that's what that was called. Is this the one that we... Cellulase. We use this one. Yeah, this is... no, it's cellulase esterase. So this is cell E. So I'm just call it cell E, because that's easier. Cell E. So yeah, this is pasted in. This should be good. So if I click save, that should do the thing. And now I'm going to do the same translate situation so that we can see that it's all protein. And then right between these, I'm going to stick a linker. And so I'm just going to use glycine. So I'm just going to look the codon table up again. Or again, I think it's probably the first time on this stream. But I'm going to look up the codon table just because I want to see what the glycine is. Okay, we're just going to go... Okay, well, so this is glycine. It's the simplest of the amino acids, and it's what I'm going to use as a linker. I'm just going to stick 5 of these in a row. And in this case, it's GG and then any of the other codons. So I'm just going to do them in order, just so we don't have something that's too repetitive. It should be GGA, GGT, GGC, GGG, and then GGT again. So you get 5. And that should, if I click enter, that should insert those there. We're going to annotate this. as linker. Save annotation, and we're also going to do the same translatey goodness. And so now we can see we've just got 5 Gs in a row, and that should do very nicely. And the reason that we have 5 Gs like this is it gives it just enough space that the two proteins should be able to fold properly. Like if you just stuck them together directly, sometimes you run into issues where, like, if part of the protein at the very ends plays into how the enzyme actually functions, or the protein functions, if you don't have a linker, sometimes they like knock into each other and they don't fold properly. But having a linker here typically fixes that problem. So yeah, that's pretty good. Fucking was making 5G jokes now. But should be able to turn that off. In theory, this should, because this is all just one protein, this should actually be done, I think. How big is this? I'm curious to know what this would cost. So let's do our usual. Wow. Okay, that's chunky. So yeah, this would probably cost, I don't know, $1,500-$1,600 to make. Just because it's kind of big. You might be able to shave a little bit off here and there, and it depends on the company that you would use. Different ones charge different amounts. But for something like this, it is quite large. But at the same time, I mean, it's not so bad. It would definitely be expensive, and it's not helpful. Like I said at the beginning, like this is not something I would ever actually have made because it just doesn't make sense. Like there's organisms that you can already use for this, and cellulase is cheap. Like also Nigel has already done the toilet paper moonshine. Like I wouldn't want to take that from him. But it's still interesting to go through the process. So I think through this you've kind of seen that, like, finding the right enzymes can be tricky. We got to talk about linkers, which is neat. We've got the secretion tag. The one thing I'm going to just add to the end here, which I don't think is present, and this is something that I've actually been forgetting to do in the other streams, is we need to add a stop codon. So I'm going to go again to my codon table here, and yeah, there's a few different stop codons. I'm going to go TAG. Also, just for those looking at this and might be a little confused, anytime you see a U here, this is talking about RNA. Since we're working in DNA, all the U's become T's. It's the difference between not tyrosine. Wow, I'm blanking on this one. They use uracils. So yeah, I'm going to just add TAG. TAG. Enter. And I'm going to do a translation. So yeah, now you see the little star. So that's the actual stop. So as the ribosome is reading this, what it'll do is eventually it'll hit, like it'll be making protein until it gets to right here where you hit tag, and it's like, oh, okay, that's stop. And so it lets go. Also, the point of this terminator, which is the thing that you always have at the end of a gene, is it's basically just a long string. Excuse me. It's a long string of like stop codons and other junk to basically force the ribosome to let go. In fact, just to show you this. So yeah, you'll see there's a whole bunch of different stop codons in here. Part of it is just to like literally make sure the enzymes let go on purpose. And sometimes there'll be other little bits in here. I don't know what this mic tag is for. No idea. I put the stop code on here, so this shouldn't get made. This should just be ignored. But yeah, I think that means we've done it. We've got the glucosidase, so that'll actually break the cellulose into individual sugar units, and we've got the cell E, which is supposed to break the cellulose into little bite-sized chunks so that the other one can actually do the work. So in theory, and we've got the secretion tag. So in theory, this should be all of them. Thymine, thank you. This is one of those things. Like when you do biochem, like they have you just memorize all of these things, like every amino acid and the structure of all of them and all the bases, and it's just like if I ever need it, I'll look it up. Like, it does make it seem like or like, you may seem a little forgetful if you've got to look it up, but like there's literally no shame in understanding that like there is just too many things to know in biochemistry to ever be worth trying to memorize all of it. You'll never manage it. It's a ridiculous amount of stuff. Just so as a refresher, the thing that, for anyone who's just tuning in, the thing that we've actually just finished is designing a piece of DNA that would, based on the prompt, and this was our prompt. It said, "Real toilet paper moonshine." So yeah, based on the prompt, we had to design a piece of DNA that would do that thing. So if you were to put this piece of DNA into brewer's yeast, it should secrete these two proteins, which are actually one big protein that are stuck together, that will break down cellulose and then ferment it. And so basically what should end up happening is you would basically end up with toilet paper moonshine. Like, you could just like chuck a roll of toilet paper in there and just let it go, and it should, in theory, turn into alcohol, which you could then distill. But yeah, I think that means we're done, and it only took us hour 40, which is not so bad. That's pretty good. I don't know if we're gonna do another one today, but because this one actually took a goodly amount of time. But I'll in the last like 20 minutes of the stream, I'll answer some questions and we'll hang out. And I mean, just for fun, I think I might pull. I mean, what do you think? Should I pull the next topic, like for the next stream, out, or should I? No, you know what? I'm gonna wait, because if I pull it out now, there's no way that I'm gonna be able to resist like looking it up and checking. Yeah, I'm gonna make sure that I don't peek, because if I know what I'm getting into, I'll look it up and then it's not as fun. Big plus on the front. Okay. Okay, here's what we're gonna do. I'm gonna pull one out, not because I'm gonna use it, but just to give you an idea of like some of the ridiculous. Or actually, I'll pull out like 2 or 3, just to give you an idea of some of the ridiculous things that are in this hat, because I think some of the things that we've gotten thus far have been pretty tame. But yeah, let's see what some of the other ideas that I've got in here. Okay, so I've got gluten producing rice. And what's this other one? Ooh, ivory yeast. So I think this is the idea is to grow ivory. I'll pull one more. I've got a couple more. What else do we got? I'm hoping for one of the really funny ones. Cheese bacteria that taste like strawberries. I'll do one more. I don't want to spoil all of them. And again, if you've got yeah, this is ants that taste different. I've been excited for that one. I hope we get that at one point. But I'm gonna scrunch these all back up, put them back in the hat. Because I do want to. So there's a chance that these will get pulled over the next time. Shit, sorry. So yeah, these are all just gonna get folded back up, put back in the thing. If you have suggestions for future episodes or future topics, you can go over to Twitter and send them to me, and use the hashtag who's Gene. And then I'll look through them, and if they seem feasible, like all the things that are in the hat, like they might sound ridiculous, but like just judging on my own like bio knowledge and like a really quick peek online, like I make a point of making sure that I only choose things that are doable, or I think are doable. I mean, I might be horribly wrong, but I choose things that seem reasonable. So, you can suggest whatever you want, but only things that I think are actually possible will end up in the hat. But also, go ahead, leave lots of suggestions, because, I mean, there's plenty of room left in the hat, and I think I'm going to be doing these episodes twice a week. So, I think we're gonna burn through them pretty quickly. Um, and don't leave them in the YouTube. Uh, don't leave them in the comments here, because I'm like, they'll get lost. If you want to actually submit an idea, send it on Twitter, because then it's just easier for me to check after the show. Um, yeah, a friend of mine is already doing THC yeast. The company is called Hyacinth, Hyacinth Bio. They do really really awesome work. They've got like an enormous amount of funding, and they've done it. Like their yeast makes THC. They've also made one that does CBD, which I believe they're one of the first groups ever to make yeast do that. And they're working on commercialization. So they're going to be literally growing the stuff in tanks, and they're Canadian, so they'll be able to sell it. Um, okay, so let's answer some questions. Here, I'll just type it out. I wish I had the presence of mind to make like a thing, but so yeah, use that hashtag. Who's Gene? Um, not like who is Gene, but like who's Gene. Um, Here, I'll just look them up. I can spell. Hyacinth Bio. I believe this is the company. Yeah, this is them. So yeah, this is their company. It's Hyacinth Bio. They do just fantastic work. Highly suggest checking them out. They're good people. Will you try to actually do these? Yeah, I mean, so the first two that we did were these ones, which is one that does egg white and one that does milk proteins. And both of these have been funded. We did a fundraiser in the last stream, and so I've got to make some little tweaks to them, just because I'm not 100% satisfied, and they are expensive. So like I want to make sure it's perfect before I submit them. But I'm gonna go in, make a couple little changes, and then submit them, and then we're gonna get them made, and then eventually there'll be a video, and eventually they'll be released for people to use. So I'm definitely going to put them up on a site called AdGene, which is this site here. AdGene is basically a repository of DNA. So if you submit pieces of DNA to them, anybody else can buy them, basically. So they'll be submitted to AdGene, and I'm contemplating setting up a store to sell them myself, but I'm not sure. For AdGene, everything is $75. It's like everything is the exact same price. But if I start a store, I'll probably sell it cheaper, because it'll just be like DNA in a tube. But I think what I'll probably end up doing is, for the people who threw in for the funding, like I'll probably contact them specifically and like send them a sample, especially the people like Nico, who threw in just an absolutely ridiculous amount of money, which I very much appreciate, by the way. So the people who helped out, I'll make sure to get them a sample, but then if I actually do a store, it'll be more readily available. But because I want to make sure that, like, even if I sell it, like, it's going to be like, if you feel like distributing it, not in the sense of like selling it, like don't just buy a sample just so you can resell it, but like if you've got a sample and somebody like comes to your lab or whatever, and they're like, hey, that's a really cool egg white yeast that you have, like, please share. I might sell kits. I'm not sure yet. That's really changing my business model, and then I've got to hire somebody to do that, and it's a whole big thing. And I'm not opposed per se, but it is something that, at the very least, I'd probably hold off until there were a variety of plasmids to sell, not just the one. And I'd have to get somebody hired on to do that thing, and it's a whole undertaking. But I'm not I'm not opposed. Yes, add gene provides them in E. coli. So you can see it. It's on screen. It says standard format, plasmid sent in bacteria as agar stab. So the nice thing about this is it means you've got infinite DNA. Like, it's not just a little sample that if you use it up, you're fucked. It's infinite. So you just, you grow up the bacteria on the media that they need, and then you just take a little sample, extract the DNA, and then you've got what you need to work with. What do you think about Moderna Inc. targeting the ACE2 receptor using their mRNA vaccine? Meh. Very meh. I'm not super into their vaccine. I don't like the way that they're going about it. But again, not a virologist. I'm just some schmuck. Is SARS a bioweapon? Absolutely fucking not. Period. Full stop. Alright, I'm just muting you. Remember, if you're going to shitpost, you're going to get banned. Are you still doing microfluidics projects? Yeah, I do. We actually just got an SLA printer at the lab, so I've been starting to tinker with seeing if I can just like print some microfluidic stuff. I've still got some plans for doing the Shrinky Dink microfluidics. I think that was really successful. The only problem is the lab is on the other side of town, and so can't really go there at the moment because the world is shut down. But once that's unfucked, then I'll be able to hopefully do that. Daniel Lopez, what's the difference of what you just made to a cellulose breaking enzyme? Literally nothing. We made a cellulose breaking enzyme. That's what it is. That's what we made. Here, where is it? This. And actually, I'm going to rename this now that it's finished. Is this the... Should be this one. What's this? No idea. Rename to... Cellulose. Cool. Now it's finished. So this'll be... I haven't put up the... so the last one we did was... Like the last stream that we did on Saturday, I think, was this guy, where we did a penicillin making plasmid. I'll throw this up on GitHub. I meant to do it before, but I didn't. So this will be up on GitHub today. I'm going to throw the Cellulose one up on GitHub. So yeah, for anyone who wants to play with it. How do you make granulated yeast, like baker's yeast, like the stuff that comes in a package? Am I understanding that correctly? So basically the way that it's made is you grow an enormous amount of yeast, and then spin it down, get rid of as much of the liquid as you can, concentrate the yeast as much as you can, and then traditionally it's vacuum dried, basically. So you barely heat it, and you just pull a vacuum and dry it that way. That way it's basically sterile, and you're not heating the yeast, because if you heat them, you'll kill them. So that way, you can make the powdered yeast. And I've actually been thinking of doing this. So the video that I'm working on, and I wanted it out this week, but I think it's gonna be out next week. Is called "There's No Such Thing as a Yeast Shortage." So basically, the plan is, I'm showing how you can isolate yeast from various places, the air, fruit, whatever. But one of the things I've been thinking about is bringing my vacuum pump home, and making a little vacuum chamber, and then just pulling a vacuum on some yeast solution, and just drying it out that way. Yeah, so it's quite doable. It's just you need some hardware. What's your favorite food that involves fermentation? Um, that is a hard question. I mean, I mean, I love pickles and I love sauerkraut, soy sauce, miso. I mean, there's so many good answers. Honest yogurt, like it's hard to pick. I love ferments. I love fermenting things. It's so much fun. Um, Consider the Dynavix QFX fluorometer. Why? Why is that better than a qubit? I do like kimchi. Yes. Gemma, what's special about the Dynavix? Are there more home cooking videos? Yeah, that yeast video is going to be on the food channel. It's not going to be on the main channel. Because I'm going to be making an enormous amount of bread. I mean, I've been making a huge amount of sourdough over the last couple of weeks and like filming it and doing that. Can't post links. Okay, but like, what's special? What's special about it? I mean, if you post a link, the moderator should just be able to let it through. If otherwise, I can just add you as moderator for a second. But here, fucking, I have the internet in front of me. I can just look this up. Novix fluorometer. So what's different? Broader sensitivity to what? Like how? How is it more sensitive? What makes it more sensitive? Oh my god, it looks expensive. I mean, it looks like it does the same things. Double-stranded RNA, single-stranded protein. Can you ferment legumes? Kind of. It sort of depends on the legume and how you want to ferment them. Like, you wouldn't want to ferment peanut butter with koji, for example, because it will break down the lipids and it basically is like it's going rancid. It's awful. I mean, here, I'm going to go into incognito mode so that I don't expose my eBay login. Novix fluorometer. I want to see if there's one used. There is not. Okay, well, that answers that pretty succinctly. Yeah, nope. Rip. I mean, you can ferment some legumes. It depends on the lipid content mostly, and it depends what you're fermenting them with, and it depends how you're fermenting them. Like, you know, miso is fermented soybeans, but like a peanut is very oily compared to soy, so I don't know how that would really respond. Is sort of my thing. So what does a fluorometer do? So basically what you do is you take a little bit of DNA and you add a fluorescent dye to it, and then the fluorometer measures how bright that glow is when it shines like a UV light on it. And from there you can figure out how much DNA is in your solution. That's another thing. When you say make the ends correct, what do you mean by make the ends correct, Jared? Has there been an ethical question raised regarding to the head cheese project? Yes, and promptly ignored. There's nothing unethical about it, in my opinion. Do you think the DNA synthesis prices are going down? Any predictions? Yes, they're constantly going down, which has been wonderful. I could give out the patent, except I don't remember what it's called. You'll just have to look it up. Unfortunately. can you ferment dangly bits? It depends what they're made of. All right. So yeah, the rest of my day, once the stream is over, is I'm gonna be building that spider silk plasmid. And actually, you know what? Now that I think about it, I can actually show you what I was doing. So this is the spider beer plasmid version 2, the one that we had synthesized. I'm gonna close this so we got more room so y'all can see what's going on. So you'll notice it's very similar to the things that we've been making. The main difference is this AOX1 promoter instead of the TEF1 promoter, which is what I prefer. This is the silk. This is the silk part, and it's quite chunky. It was a pain to make. And then there's this mineralization sequence on the end, so that it's infusion. So like it's one big protein, and in theory, on contact with like mineral water, it should pull minerals out of the solution and make like solidify. Basically, is the plan. So yeah, but the issue is because of this promoter. I clicked on the wrong thing. This promoter. This promoter only seems to work in Pichia pastoris, which is a fucking awful type of yeast that I really do not like. So modifying Pichia for whatever stupid reason has just been very frustrating. Like just getting the DNA into Pichia has been very, very difficult. And that's probably just because I've never managed it successfully, so I don't know the exact reaction conditions that'll make it happen. Which is why I've been going over and over and over and over and over again to and just redoing the protocol until it works and just changing different things. But while we've been struggling to get this thing to work in yeast, we're like, you know what would be really great is if we could just take the silk out, stick it into something else, and then still have some silk to play with, even if it's not coming out of yeast originally. What we've been working on is cloning out this silk sequence and the mineral sequence and sticking it into an E. coli based plasmid. And so this is the thing that we're trying to make. You'll see the let's jump here. So this is the E. coli plasmid. Somebody threw in. Thank you, Hayden. Greatly appreciated. Did you do electroporation with the Pichia? No, I don't have an electroporator. I made an electroporator and I tried it, but it didn't work. So the point being, this is now the plasmid that we're trying to make, and the only reason that this was difficult was because PCRing out this silk chunk was a gigantic pain, like truly a gigantic pain. I really need to check where that primer sticks because I'm not seeing a minute. Oh, there it is. Okay, wonderful. So yeah, just getting this out was really hard. The gene itself is extremely what's called GC rich. So if you look at it, you'll see loads and loads and loads of G's and C's, and this is because the amino acids that it's primarily made out of is alanine and glycine. And if you look at the codon table here, you'll see like just shitloads of G's and C's and G's and G's and G's and C's. So this makes it both very difficult to like physically synthesize the gene chemically, but also it makes it very difficult to PCR because it's so GC rich. It's also very sticky. So the DNA literally sticks together too hard and it doesn't come apart properly during the PCR cycle. And it's very repetitive. So the primers will sometimes stick to totally random, incorrect places, which is very annoying. So what I ended up doing was, so I ran the PCR reaction 3 times and it didn't work every single time. And that was very annoying. So what I ended up having to do was going over to the spider beer plasmid. And if I jump to right around here, So you'll see BAMHI. This is a restriction enzyme. So it's an enzyme that if you add it to the solution will cut. You can see it on the screen where the exact spot where it cuts. So I cut with BAMHI, and then there was one near the end, which I should be able to jump. Yeah, AGEI. So I had to actually cut this, and literally like cut this chunk out that I wanted, just so that I could then run the PCR on it. And something, I don't know why this fixed it, but it did. So something about cutting the chunk out seems to have exposed an end. Oh, thank you, Nico. So it seems to have exposed an end that made it much easier for the PCR to work. Again, I don't understand why this fixed it, but it did. So big shrug. I don't. This is bio. Sometimes things just work for reasons you don't understand, and it's just the way that it is. Okie dokie. So yeah. So basically, what I had to do, I did that. So once I managed to get this fragment out, which I did, now what I'm going to do is I'm going to this plasmid started obviously without the silk in here, and the other thing that it didn't have, which hopefully I can just scroll up and find it. Did I scroll right by it? Yeah. Okay. So then the other thing that we had to add was a secretion factor. So in the same way that we've been using alpha mating factor before the yeast genes, E. coli have the same sort of thing, but they've got the secretion factors are much smaller. So in this case, it's just this short little thing. So we made this with like PCR primers. So we basically like bought half and half, and just had them like stick together. And when I make the video on this, I'll talk about this in detail. Yeah, so we made this piece, we extracted the silk, and we have this backbone, which I cut and basically made a hole to fit this stuff. And so then later today, I'm going to mix all three pieces, and I add a combination of enzymes that does a process called Gibson assembly. So it's a whole bunch of different enzymes that will basically stick all these pieces together in exactly the right way, hopefully. And then once I've done that, I can then take the finished plasmid, put it into E. coli, and in theory, if it's assembled properly, I'll have silk tomorrow. So that would be really, really, really great. So we're making progress, and I'm basically just like not rushing, but like I'm trying really hard to work my way through this and get it done so that I can make a silk update video. So I can be like, we've made some silk, it's done. Now these are the other problems we're now working on, because like I don't like doing little short update videos if there's nothing to like show for it. Yes, we have the DNA, whoop-de-doo. If I can't get it to work, it's useless. So with this, I can actually show some actual function. And for those curious, I think I've actually made some silk by accident already. So the spider beer plasmid is built to be copied but not work in E. coli. So you'll see this AMP-R and ORI. These two pieces let this be copied in E. coli, but not actually run in theory. Except that the kind of E. coli that I have are called NEB turbo cells. And for whatever reason, and I don't understand this, and I've talked to some friends about it who also work with turbo, turbo are weirdly leaky in the sense that they kind of don't respond to promoters properly. So if this was in yeast, the only time that this gene should run is when this promoter detects that methanol is present in the media. Except that turbo is leaky, so it just kind of runs things. So I think what ended up happening was it actually ended up running this gene, like running the program, even though it wasn't supposed to. The reason I think this is because I've grown a lot of E. coli over the years, like a lot. And they always look the same. Like when you've got them in a tube and you're growing like a mill's worth, like they look the same. It just looks like a kind of, like it's a little pellet. It behaves the same way. If you shake it, it disperses really easily. But when it had this DNA in it, and I tried putting it in turbo cells, the pellet was like goopy. And like even the colonies on the plate when I first did the transformation, when I like poked at it with a loop, it was like weirdly firm. And again, I've handled a lot of E. coli over the years. I've never experienced this before where like you poke the colony and it's like firm. I had to like really scrape at it to get it off the plate. And the pellet of cells when I grew it overnight in a tube was like stretchy. Like I could not disperse it very easily. It was like goopy almost. Like it would like spring back and forth before I had to like really shake it to get it to disperse, which was very strange. I've never seen this happen before. So the only thing that I can think is that the turbo were leaking and accidentally making some silk, which is weird. Very weird. But this gives me hope that now that I'm building this E. coli version, which is like actually meant to run an E. coli, it should run a lot faster. It'll have the secretion factor that's an actual like E. coli secretion factor, so it should actually like come out of the cells properly. Oh, Martin, that is a good point. Can't believe I didn't do that already. I will do that. Thank you for the suggestion. Anyway, so this has a bacterial secretion factor. It's got the silk. It's got the thing. So it should run, ideally. And so basically, I'm just gonna like poke at the colonies, and whichever ones are firm should have the right gene in it. And then I'm gonna take a little sample, do a DNA extraction, make sure it's correct. So yeah, that's kind of where I'm at. That's the rest of my day. In theory, like if you follow me on Instagram, in about 2 days, if you see me posting pictures of champagne, then it fucking worked, and I've made some silk. I've been working on this silk project for literally years, like years and years and years and years. I'm so excited to finally almost be there. Like this is one of the reasons I spent so much time yesterday just doing this, because I'm so damn close. I don't like using E. coli. This is true. I fucking hate E. coli. They're gross. But they are very easy to work with. So if this is why this was a last resort, like it basically, it's one of those, like it cost us almost nothing to make the primers that lets us build this plasmid. It's a plasmid we already have. The secretion factor cost almost nothing. So it's only a stopgap. It's just to get some silk made so we can start actually experimenting with it. Also, my friend is going to be hopefully sending me a different kind of bacteria to work with. It's a species of Vibrio that grows twice as fast as Turbo. Like Turbo are already very fast for E. coli. Like they're you can get a colony on a plate in 6.5 hours with Turbo, which is just ridiculous. Vibrio, you get colonies in 3 hours, which is crazy. That means you can do like multiple cloning runs per day, which is insane for biology. Like biology is inherently slow. Like the fact that you can do all that work in 1 day is just nuts. I wouldn't have the energy for it, but it is possible. So the nice thing with the Vibrio is they use all the same promoters. Like all the promoters that work in E. coli work in this species of Vibrio. So I should be able to just transfer all of this work. So once I've made this spider silk plasmid, I should be able to just very quickly switch to the Vibrio, stick it in the Vibrio, and then like that's actually a species that I'd be much more willing to work with. And hopefully it doesn't reek in the same way that E. coli does. And if it doesn't, then it's just going to be my go. Well, I mean, even if it is, like it's going to just be my go-to species to work with because it's just fantastic. So yeah, that's the plan for the rest of my day and the rest of my week, really. It's all silk all the time. It's silk stuff, and then I'm gonna be yeah, it's funny. People are like, you're gonna be rich if you make silk. I'm releasing it for free. Why? It's not the point. The point is to make something really cool and share it, because that's my thing. I don't want to be like a baron of owning silk company. Blah, fuck that. I want to make something cool that people can play with. There've been a lot of requests for a Discord. I'm unsure about how I feel about that. I'm not big on Discord. I'll think about it. There was a bunch of other questions that I missed. Lactose questions will be ignored. Could it be that your promoter is broken? No, the promoter should work fine. Turbo is just leaky. And like I noticed this with like literally a dozen other plasmids. Well, thank you, Andreas. I've noticed this with like literally a dozen other plasmids with Turbo. Like they just leak. All of my fluorescent protein plasmids, for example, that are built to work in E. coli, should only run if there's lactose present in the media. But in the turbo, every single one of them ran no problem. Whereas in BL21, which is a different type of E. coli, not a single one of them made even a whiff of fluorescent protein without the lactose. So it's just a weird thing with turbo. Like something about them is just inherently broken, so they just run DNA even if they're not supposed to. Why is the THC produced by your friend only made inside the cell? Because there's like 7 or 8 proteins, and they require chemicals that are inside the cell to function. And so if you try and export them, it's just not going to work very well. Like you're losing efficiency, and they'll get degraded by all the stuff that's floating around in the media. Detection of coronavirus. I am working on a video already. I have all the materials in my fridge. I'm just waiting on more materials. I don't want to just do one test. I want to really make sure it's a comprehensive video. I'm going to do a PCR-based test. I'm going to do a lamp-based test, hopefully, and ideally an antibody-based test as well. Yeah, so that's that. What else we got? Asking if the liquid turned blue. Yes, actually, the liquid did turn blue. I did actually manage to make lactase, so that's good. I was just letting it grow more so that the activity is higher, because it took about a day for it to turn blue. I would like it to turn blue in about 10 minutes. But I mean, I've already got the proof that it works. I want to see if I can get it faster, which should just be a matter of letting it grow longer. Can you emulate cells? No. Unless you've got like a supercomputer or 4, no. There's hundreds of thousands of things going on in a cell at any given time. You'd be simulating literally billions of molecules and atoms all interacting, and just simulating one protein is an enormous computational task, not to mention literally hundreds of thousands. The only way is to just do it. There's no simulation. There's no compiler. You just run. Do you have a recipe for Xgal? I bought Xgal. I don't mess with that. It's not expensive. It's pretty cheap as these things go. So I just weighed out the correct amount, threw it into some DMSO, let it dissolve, called it a day, and then just like chuck that into my test solution. If you got infected, would you have to scrap some of your projects? I'm not going to get infected. Period. I'm a crazy paranoid biologist. The chances of me getting infected are probably lower than just about everybody else. Everything is sanitized on the way in and out. I wear like a full respirator when I leave the house. Like I don't touch my face. Like, I mean, this is the other thing when people are like, "Oh no, I'm gonna touch my face." Like I'd never realized how much I touch my face. I'm like, y'all, I'm a biologist. I've been doing micro and just bio for years. Like this idea of like don't touch your face. If you're handling E. coli, of course you're not going to touch your face. So you already get into the habit of don't touch your face. So a lot of these issues just not things that I'm really worried about. Like literally everything gets sanitized on the way in. I keep a bottle of like sanitizer at the front door. Like there's a hook. Like nothing crosses my threshold if it has not been fully sanitized. Okay, what else we got? Okay. And I mean, I realize I've touched my face a whole bunch of times on the stream, but I'm also not in a position where I like when I'm aware of the pressing concern, like as soon as I leave the house, like my hands are in my pockets the entire time, like unless I need to interact with something. You're young and live alone, so what if you get it? Well, it's basically lung herpes. Well, it's not lung herpes, but it's not something you really want. Shortness of breath, like the amount of shortness of breath that people get from it is just terrifying. Like it's not something I want. It is not something I want to have to deal with. Is folding at home the future of finding drugs? No. That's folding at home is neat, but drug discovery has many tools already. There's already great tools where you can just like make up compounds and it'll tell you which things it's gonna stick to. Also, like most of like drug discovery is done combinatorially, so like they'll buy 7 or 8 like precursor compounds that are just easy to stick together, and they have a robot make like 200,000 different compounds, and then they run all of them on an assay against the thing that you're trying to like find a drug for and see what sticks. Like it's there's literally billions of dollars in drug discovery. Like folding at home is child's play comparatively. It's neat. Don't get me wrong. It's a useful, very cool thing. It's not the future of anything. What else we got? Okay, I'm gonna do another like 5 minutes of this, and then I need lunch, so we'll call it for the day. I'm gonna be doing the next stream on Friday probably. So I hope all of you tune in. I think this I don't know about you, but I'm liking this format. I think we've already made a whole bunch of really cool pieces of DNA. I'm hoping that the next one of the next ones that we pull is something that's actually fun to synthesize. But I mean, most of the ideas on here is not something I'd actually ever make. But yeah. You're getting hidden. Please take your conspiracy theory and fuck off. Okay, what else we got? What would it take to make a spider web out of your silk? So this is actually something that I've thought about, not a spider web particularly, but like just how you make fibers. So once I've got the Friday 2 p.m. Eastern, same as always. Anyways, so once I've got a tank of silk, basically, the first thing is I'm gonna have to extract the silk from the solution, which is part of the reason why I did the excretion stuff. So from there, then you actually have to dissolve the silk, and there's a few different solvents that you can use for this, but the one I'm probably going to use is 9 molar lithium bromide, just because apparently that's the thing that works really well. So you take your silk solution, you mix it with your 9 molar lithium bromide. Well, I mean, what would probably be more likely is I'll save the supernatant, which should have the silk in it from whatever I'm growing it in. And then I'll use ammonium sulfate to precipitate the silk out of solution so that I end up with like a solid mass of silk. And then I'll dissolve it in 9 molar lithium bromide, at which point you have to dialyze the solution to remove all of the lithium bromide, which basically you put it in a bag full of you put it in dialysis tubing and soak it in water and just change the water, a couple times a day. And then from there, you have a really concentrated silk solution, which you should be able to inject into a bath of probably sodium hydroxide and methanol, or maybe isopropanol, which is what's called a coagulation bath. And from there, you should actually get fibers. And once you've got fibers, you can do whatever you want with them. Okay, what else we got? P2A is not a secretion sequence. P2A is a cleavable linker. You're getting muted because no shit posting. What else we got? After the virus, will you continue this style? Well, the virus is going to last 1.5 years at the bare minimum. So I'll have been doing this for a while. I don't know if I'm going to continue doing this as regularly as things open back up, but I really do like this format. It's been a lot of fun. I think y'all are learning some stuff. I'm learning some stuff. It's been really great to practice. We're making a bunch of really cool DNA. So yeah, I'm definitely going to be streaming more. I don't know if I'll necessarily always be streaming this. I want to do some streams where I'm actually like working. And like, maybe at the bench. I'm going to be doing some like food streams as well. But for the time being, I'm going to start with this just because I really like this format. And I think it's been kind of fun. All right, one or two more questions, and then I'm going to call it for the day. Any physics type videos coming up? Let me look at my list. So the most physicsy one that I had planned, but the problem is like the whole world shut down. So like my ability to do physics videos is really limited. Like I've got my biolab in my house, but like I don't have the ability to do a whole lot of physics stuff. The next physics video that I have planned, because it's all stuff that I have here, is going to be measuring cosmic rays using the same gamma ray detector that I used in my last video. So the plan is we're going to measure cosmic rays, muons specifically. So we're going to measure both their counts. So we're going to use like a directional measurement system. So we're going to basically measure the count of muons as you tip the detector over. And so this is basically a simulation of if the muon had to travel further. And so you should see a drop off, like a very specific drop off in count number as you tip the thing over. And the reason that it's a very specific number is you can then use that the graph, like the curve of the fall off of these particles, as a way of measuring time dilation. So the muons are very short lived, and so, but the only reason that we even detect them at sea level is because of time dilation. So we'll be able to measure that. We'll also measure a muon decaying in the detector. So a muon decays into an electron and some other stuff, but the electron's the thing that we can detect. So we're gonna so you basically, and I've done this. I know it works. You see this gigantic spike from the muon because it's got more energy than literally anything else you'll ever detect. And then you see this second little spike within 2.2 microseconds. That is the actual electron. So it like decayed within the detector, and the decays are very rare. Like I left the thing running for 8 hours or 4 hours, and I think I got like 4 detections. Like it's really, really rare to catch this, and I was using a massive detector. So yeah, that's gonna be pretty cool. But yeah, that's one physics video. I've also got the synthetic opal thing that I've been working on for a while. The only thing that's slowing me up on that one is I need to run the experiment again, which means access to a chem lab, which is currently restricted due to the fact that the whole world shut down. So yeah. Is it possible to move forward or backward using time dilation? Forward, you can always move forward in time. What else we got? Yeah, so I'm gonna do one more question and then I'm calling it because it's. I mean, I gotta get to the rest of the stuff I'm doing today. Somebody asked, what would you describe this if you were trying to learn for looking it up? Just look up muon time dilation experiment. Should be fun. It should come up. There's a few papers. All right, I need one more good question, and then I'm gonna call it. What else we got? No, nothing. What is the worst part of genetic engineering? Okay, that is actually a really good question. So easily the worst part about genetic engineering is it's not code. Like there's no compiler, there's no debugger, there's nothing to tell you why a thing didn't work. Like you could design a thing and you think it's perfect, and but then you end up spending like 6 months trying to figure out why the hell it doesn't work. And at the end of the 6 months, you might not even know. Like you might have figured it out. You might have run every test you can think of, and maybe you figured out why it didn't work. But otherwise, you might be shit out of luck. And so you just have to completely redesign the thing, and you're just like, you have no idea why. Like the PCR that I was struggling with, I have no idea why cutting the plasmid fixed it, but it did. So yeah, that's sort of the hardest part. It's the not knowing. It's you spend all this time and effort and money designing a thing and buying all the supplies, and then sometimes shit just does, and not even sometimes, most of the time shit just doesn't work. So like this is why that I'm doing this stream, because if I can basically convey the idea that it is so much better to just buy a thing pre-made, like pay a company to just make your plasmid for you. Don't do subcloning. Subcloning is awful. If you can avoid it, don't do it. It's expensive. It's a pain in the ass. Nobody likes doing it. Shit fails all the time. You end up burning time and reagents. It's just not fun. If you can pay a company to just make a thing for you, it is so much better and you get so much more done. Andrew Hill, thank you very much. Greatly appreciated. And you know what? The hardest part really is just being able to grit your teeth and like get it done. And like understand that failure is part of it, and you're just gonna have to suck it up and like deal with it. Like it fucking sucks. There's no sugarcoating this. Like when I did a little bit of teaching at one point, and we always talking about how like the fifth week, because by week five you're really like into the thing. By week 5 everybody's in tears, like nothing's working, everything's broken, everyone's just like a crying mess, and everyone just like goes out and gets really drunk and then comes back the next day and they're like, okay, we can do this now. It's just the way it is. Like it's gonna kick your ass, it's gonna suck the entire time, but when you finally make it all the way through and your thing works, there's nothing better. There's nothing better than making it through something that is frankly ridiculous. Is you like spitting in the face of nature and going, no, goddamn it, you're doing this the way that I say so. And you make something ridiculous and wonderful, and it can be inspiring, or it can be just wonderful, or it can just be stupid and fun. And getting to the end of it is just worth it. It's worth sucking it up and dealing with the difficulty. Like there's one of my favorite YouTube channels is Exerbia. There's a quote from one of his videos that I really like, which is, in order to shit fire, you must first chew glass, which is anything worth doing is very, very hard. It's just the way that it is. But yeah, so that's the hardest part about genetic engineering. It's the not knowing. Okie dokie. Well, so that was going to be my last question. It's 4:30. We've been doing this for a little while. Our plasmids have been made, which, for those wondering, this is if anyone who's just tuning in, this is the thing that we made today. And this will be posted on GitHub just as soon as I've had time to eat lunch. And yeah, so thank you very much for stopping by. I hope you guys have I hope you guys have enjoyed. This has been a lot of fun. I'm looking forward to doing this again. I will be doing the next stream Friday, 2 p.m. Eastern Time. I hope to see all of you there. And yeah, stay safe, stay happy. Try not to go too stir crazy. Stay healthy. Clean all of your materials when you come in the house. Wear a mask. All these things. Have a good day. Try not to stress too much. It'll be okay. And I'll see you next time.